Welcome, ladies and gentlemen, to this series of lectures on membrane potentials, a build-up to nerve tissue histology and anatomy, histology and cell physiology. The objectives of this series of lectures is to be able to help the students be able to explain the role of electricity in body communication. Also, the student should be able to describe the origin of the resting membrane potential or voltage and be able to discuss the mechanism of how action potentials are produced and as well as graded potentials, and then draw differences or contrasts between graded potentials and action potentials in terms of their generation as well as their functions, and be able to illustrate action potentials in terms of how they are conducted or transmitted for signaling. A quick introduction. I think we've already covered the principles behind the generation of electricity in biological systems. And the background is that with the exception of water, the major chemical substances in the extracellular fluid, it's sodium and chloride, while in the intracellular fluid, the major electrolyte is potassium. So electricity results in biological system results from the distribution of these major electrolytes across the cell membrane. This electric phenomena plays a role in how cells integrate the system's functions and how electricity gets to be used by the body in signaling or communication. Electricity in biological systems as well as in non-biological systems exists as a result of the interaction of electrolytes, how these are able to flow, and that would be the amount of flow would be called flux or the current, and how this can do work. And it's as a result of the fact that these particles will not be able to move unless there's a force that is moving them or attracting them. And this force exists between like particles or unlike particles. So the basics are that when charge is separated and these charged particles are on the opposite side, the force that will exist will actually bring them together. This potential created is called the electrical potential. And also this potential is also called the potential difference because it is determined by the difference in the amount of charge between 2 points. And if you remember your basics in physics, you remember that this potential difference is what we call voltage and is measured in volts. Basics continue, so the movement of electric charge is called current. Basically, that's the amount of charge that flows, or the flux. And so the electric force between charges tends to make them flow, producing a current. If the charges are of the opposite sign, the current brings them towards each other. But if these are like charges, the current increases the separation between them because of that repulsive force. So the amount of charge that moves, in other words, the current really, or the flux, depends on the difference between charges in terms of the potential, and also the nature of the material across which this current should flow. Okay, so the hindrance to the flow of charge was explained by Ohms, and he said it is called resistance. And the relationship is that the more resistance there is to the flow of charge, then there will be less current flow. But the current is directly proportional to the potential difference. So the larger the potential difference, which is determined by the difference in the amount of charge, the larger the current is likely to flow in that medium. So Ohm's law then states that the current is inversely proportional to the resistance, while it gets directly associated with potential difference or the electric potential. So materials that have a high electrical resistance will be called insulators, whereas the opposite material that is able to allow flow of charge will be called conductors. And then you realize that biological membranes are made up of fat and they have less electric charge that is able to flow across them. Therefore, we say membranes or biological membranes are insulators. Unless there's a channel that allows that flow to take place, then we'll have that conduction taking place. So the intracellular fluid and extracellular fluid will contain numerous ions or electrolytes, and these can actually carry current. So when they flow, the amount of those electrolytes flowing will be called the current or flux. But the lipid biomembrane is a region of high resistance because, remember, it is made up mostly of the fatty tails of the phospholipids. Therefore, the intracellular and the extracellular fluid are the areas of low resistance. So in the water environment, the aqueous environment of the extracellular and intracellular fluid, charge is able to flow, and that will allow current to exist. So there are a number of scholars who have, or scientists who have done a lot of research on the electricity in biological systems, and we have some of those include Nasty and also Goldman. And so Nasty developed an equation that describes how we can calculate the electrical equilibrium for each of the electrolytes in the biological system, like potassium, chloride, and sodium. So he describes that equilibrium potential for ions actually exists. That is the electric potential necessary to balance the movement of these ions as a result of their concentration difference, but also as a result of the fact that there will be an electrical attraction or repulsion that is also causing the movement. So ions will move because of concentration difference, but they will also move as a result of the attractive or repulsive forces. So where these forces balance, the movement as a result of charge difference and the movement as a result of concentration difference, that is called the electrical equilibrium. And we have an equation devised by Nast that can actually be used to calculate the equilibrium potential for each of the ions. So the membrane potential depends mostly on the intracellular and extracellular fluid concentrations, as we have alluded to. And we are focusing on potassium, sodium, and chloride because these are the major ions in our biological systems. But also the equilibrium potential depends on how easily it is the electrolytes for them to flow. And so the relative permeability of these membrane ions also tends to affect each of the equilibrium potentials. You find that potassium easily leaks across the membrane. Why? Because the potassium channels are almost always slightly open. So the permeability of the membranes to potassium is higher than sodium and potassium. So the equilibrium potential for potassium is very different from sodium and chloride. So this is what we are trying to communicate here. And so then Goldman comes and talks about the fact that each of these electrolytes, whose electrical potential has been calculated by Nast equation, can actually contribute to the total membrane potential of the cell. And so we also have this now, the Goldman equation, that helps us to calculate the total equilibrium potential contributed by each of the ions. And this brings us to now the resting membrane potential, which is the sum total of the membrane voltage across the cell membrane that is produced by the individual ions, total together as equilibrium potentials produced by these major electrolytes. So all cells under resting conditions will end up with a potential across the cell membrane, which by nature will orient in such a way that the inside of the cell is highly negative while the outside of the cell is highly positive. So this is called the resting potential. Why? Because this is the potential across the cell membrane there when the cell membrane is not under the influence of any stimuli. So it is at rest. So there are devices that we can use, micro electrodes that can be used to measure the voltage across the cell membrane. And then we will find that actually it's a small voltage. And so because it's a small voltage, we will not measure it in volts, but millivolts, 1,000th of a volt. The conversion, by conversion, the extracellular fluid and intracellular fluid is considered at a voltage of 0 because in those media, extracellular and intracellular compartments, the positive and negative charges, they cancel each other. The excess positive and negative ions come to align across the cell membrane to be electrically attracted to each other. So the extracellular and intracellular fluids are voltage 0. So the polarity or positive negative of membrane potential states in terms of what? The sign. So you always find that the membrane potential have a sign. If it says positive, then it means there are these excess positive charges inside. If it says negative, then it means there's excess negative charges inside the cell. So the magnitude of the resting membrane potential varies from about -5 to -100 millivolts depending upon the type of cell. So you find we've noted that, for example, skeletal muscles generally have their resting membrane potential at -90 millivolts, smooth muscles at -55 millivolts, and erythrocytes, red blood cells, at about -9 millivolts. The nerve cell or neuron about -70 millivolts. So the membrane potential of some cells can change rapidly in response to stimulus. And this ability becomes very key in their function for such cells. So from here, we will begin to look at how are the membrane potentials generated. And this is basically as a result of the fact that there exists a tiny excess of the negative charges inside the cell and excess positive charges outside the cell. So these excesses inside electrically get attracted to the excess positives outside and vice versa. Thus, the excess charges collect as a thin shell tight against the inner and outer surfaces of plasma membrane. This therefore creates the membrane looking like or behaving like a battery, which is got a positive terminal on the outside, a negative terminal on the inside. So basically this is what we are trying to say, and this illustration is based on the major electrolytes where we have more sodium in the extracellular, more potassium in the intracellular, and more chloride in the extracellular. So what are the factors that determine the resting membrane potential? Basically, the magnitude depends on the difference in electrolyte concentration, but also on the electrical potential, which basically is the equilibrium potential as a result of the concentration difference and electrical difference caused by attractive and repulsive forces. So also the difference in membrane permeability for each ion. So you find that there is more permeability of the membrane to potassium, and so potassium will have a way different membrane potential, and it will determine. So potassium is the main determinant for resting membrane potential for most cells because of its high permeability. And then we also talk about the fact that these concentrations of sodium and potassium, they are very, very important, and they are created or maintained by the sodium-potassium ATPase or sodium-potassium pump that pumps 3 sodium ions outside for every time it is pumping 2 potassium ions into the cell. So these illustrations are just showing that if we have in compartment 1 sodium chloride and compartment 2 potassium chloride separated by a membrane that can only allow potassium to flow, then what we expect is that potassium will be able to move into compartment 1 as a result of a difference in concentrations. But eventually this will be opposed by the fact that the potassium begins to move into compartment 2 as a result of the attractive forces to the chloride ions. So this is the illustration of creation of membrane potentials as a result of concentration differences. And so basically this is what we are trying to illustrate here, and eventually all the way. And so then you are able to calculate using the nasty question, that is the summary of the equation, and then the total resting membrane potential calculated from everything else. The Goldman equation, and that's the end. And then we will look at action potentials and graded potentials, the changes in the resting membrane potential. See you in the next lecture.